You can hear me, right? I'm audible. Yeah.
I can. Can you hear me?
Yes, sir. So I will, before we start, I will just, share biodata of our speaker for today's. Thank you, sir, for accepting our invitations in spite of your busy schedule. And we are very fortunate to have you as a speakers on this day. And also, I would like to thank our VC, Honorable VC, for organizing such important and interesting webinars. Uh, our today's speaker is Dr. Rajan Sriraman. He is a biotechnologist by training, and he has done his master's from MS University, Baroda, and he did his PhD from TERI, that is Tata Energy Research Institute from Delhi, and he did his postdoctoral training at the Technical University of Aachen, Germany. He has been working in vaccines and R&D for the last 16 years in different organizations and roles in, and he has played a very vital roles in India and Germany. He was part of the team that developed world's first typhoid CRM-197 conjugate vaccine. And he played a pivotal role in the development of pneumococcal conjugate vaccines, hepatitis A and measles vaccines in his long career. And he has extensive knowledge on HPV vaccine. He was trained at NIH on pseudovirus neutralization assays and has supervised several master's and PhD theses on this topic. And he has authored 24 peer-reviewed papers in international journals, and he has filed over 14 patents in recombinant protein expressions and vaccine technology. And he has also authored 2 book chapters. And he is currently the vice president, and he is heading the vaccines R&D at the Reliance Life Science Mumbai. Thank you, sir, for accepting our invitations, and it's a privilege for having you here. You can start your.
The pleasure is entirely mine. I should profusely thank Professor Sambasiva Rao, who this was just a chance call that led to this webinar. Thank
Morning,
you,
Rajan. I am here.
Yeah.
Yes, sir has
so, yeah, thank you for this invitation, and it's a pleasure talking to you. And since I have a sudden commitment coming up at 12, I thought I would try to close it as early as possible, which is about 11:50 or 11:55 at the max, because I need to log off and get on to another call. So I hope you you accept my apology for that. So without further ado, I'll just get started with the HPV vaccine, which is also a cervical cancer vaccine. I hope everybody can see this.
Yes, sir.
And if you have any difficulty, please do let me know. Right. So what I'm going to lead you through is a cervical cancer history, the history of a vaccine development. In the current situation is particularly important because the whole world knows that we are struggling to generate a COVID vaccine. So you would see how long actually it takes to come up with an effective vaccine, and what are the institutions and departments that get on board to come up to address any public health challenge. So this talk is essentially something that starts with the basic science, and then it translates into clinic. And of course, with all the hard work, you definitely need some bit of luck, which is serendipity, and then you would see there was a serendipitous discovery that led to an interesting vaccine coming up. So this is how I have outlined my talk. First, I would be, in first part, I would be talking about the natural history of HPV infection and HPV and its role in cervical cancer. And the last is how do we develop a vaccine for that. Now, coming to the natural history, you would appreciate that any cancer is essentially deregulating the cell cycle. I mean, the cell cycle per se is an extremely well coordinated and controlled mechanism, wherein you have growths. There are two growth phases, and a synthesis and a division phase. And this is all textbook knowledge. But what is essential for this talk is there are several critical checkpoints in the cell cycle, and two of them of importance for us is controlled by P53 that corrects any error in DNA replication. So from when the cell enters from S to G2, the DNA has to replicate, and the P53 then corrects any error in that. And in the G0 arrest phase, G1 to G0 arrest phase, there is a protein called retinoblastoma that binds to elongation factors and keeps it in the arrested phase. So these are two cell cycle check. So we'll see how this progresses. Now, coming to cervical cancer, it's about the fourth largest common cancer in women. The infection of human papillomavirus is etiologically linked to all cervical cancers, and infection spreads by intimate contact. The onset of the infection happens because of sexual debut. The infection rate increases sharply by the age of 25 to 30, and it plateaus by 40 to 45 years. Nearly 56% of all cervical cancer death occurs in developing countries. Periodic screening of cervical cytology helps, but it is, as we will see in this talk, it's extremely cost intensive in terms of public health spending. Now, if you were to look at the disease burden, this is a virus that is across the world. It is present across the world with varying degree of severity. The dark shade being highest burden. India is probably the second largest in terms of disease severity, which is, I'm not sure if you can read this. This is about 11 to 10 cases, 17 to 10 cases per 100,000. Whereas in less developed country, and especially in Africa, this is also a region which is riddled with HIV infection. The rates are extremely high, which is about 25 to 26 cases per 100,000. So what also this tells you is that if you look at the spread, the burden of the disease is also linked to how the governments are spending on the public health. So the distribution of disease is directly linked to the GDP spending on public health. Now, let's go back some years ago and see how the disease came to be identified. In about 50 years ago, in the United States, about 32 or 33 cases per 100,000 were present, meaning about 33 women per 100,000 would report cervical cancer, which is even worse than what is there in Africa as of today. But by 1954, there was a pathologist, Georgios Papanikolaou, who developed a very simple tool with which we could identify precancerous and cancerous cell by simple microscopy. So if you can see, there is a method by which you could identify if the nucleus of our epithelial cell is getting degraded. Needless to say that there are two factors that are critical to this. A, you need a trained cytologist at a primary health center to be able to perform this screening population-wide, and also the cytologist must be sufficiently experienced to identify the deregulated nucleus order. However, this particular screening strategy was introduced in Scandinavian countries way before the cervical cancer vaccine came about, and they saw a significant reduction in the cervical cancer burden. So therefore, what is critical is there is a tool. If you could implement those screening tool, probably you could reduce the disease burden. Now, coming to cervical cancer, what was the patient profile? The high-grade lesion was observed in people who were in their reproductive age, who had an early sexual debut and had multiple partners, poor sexual hygiene, smoking, alcohol, and chlamydia infection. Mind you, these are all indications that are seen in about 50 to 40 years ago. Therefore, looking at all these profiles, the disease was classified as a sexually transmitted disease. That this is something that is linked to intimate contacts. Now, there was another set of observation that led to the discovery of the causative agent, that there was an observation that the rabbit and cow skin warts also become cancerous. And when they looked at the cell cytology and cell morphology of these cancerous tissues, they looked very similar to what the Papillomaviruses stains would reveal. So, and these warts were known to be caused by rabbit and bovine papillomavirus. So therefore, it was a direct or indirect indication that the cancers are actually caused by human papillomavirus and not any bacterial infection or herpes infection. In the 80s, Professor Zurhausen from a German cancer center employed in situ hybridization tool to screen different warts. And what he found was, when he looked at the plantar wart, which is a very common wart most of us would have had in our feet, and then we put those band-aids to remove the cones. This is basically HPV-1. That was the first HPV identified, and therefore called HPV-1 in 1985. And from then on, there were a series of discoveries. The first, the mucosal HPV was HPV-6, followed by HPV-16 and 18. HPV-1 to 5 are all involved with skin lesion. So what Zurhausen concluded is, no matter which wart he was looking at, every single wart contained HPV. So clearly there is an etiological agent which is involved in all wart lesion. So if you were to look at papillomavirus today, the papillomaviruses are present from bird species onwards. So there are avian papillomavirus, there are reptilian papillomavirus, there are mammalian papillomaviruses. And therefore, the evolutionary history of this virus is actually close to millions of years old. So this is a virus that has evolved with us. It's not something that has come up now. And it's a DNA virus, so it evolves very slowly. And out here, what you see is high-risk papillomavirus, which is the papillomaviruses are classified as high-risk and low-risk. If it is infecting the mucosal surfaces, it is called a high risk because it leads to cancer. And cutaneous papillomaviruses are low risk because they generally are forming benign tumors and warts. And as on date, more than 200 types of different HPV is known, and all these typings are done based on L1 nucleotide sequencing. We will come to this L1, L2 business in a short while. Now, okay, so there is a definite link of papillomavirus causing cervical cancer, but in addition to the cervical site, the other mucosal sites are also known to have cervical cancer HPV infection, like oropharynx, anus, oral cavity, larynx, vulva, and penile cancers are also to some extent caused by HPV. And if you look at the disease mortality, the disease mortality rate that is contributed by India, it's about 60,000 cases per year, and that's significantly high compared to any other country in the world. Now, since I said there are about 200 different types of papillomaviruses, not all of them are involved in cancer. So of the 200, about 7 are predominantly involved across the geography in causing the cervical cancer. So if you look at the disease caused in Brazil, Mexico, India, China, almost more than 90% of cervical cancer is caused by just these 7: HPV 16, 18, 31, 33, 45, 52, and 58. And although there is a regional discrepancy as to which of these types are involved in causing higher or lower burdens of cervical cancer, but by and large, these are the 7 types that you could appreciate across the world. Now, having seen that HPV is linked to cervical cancer, let's look at what is this virus about. This is a DNA virus, has a genome size of a very small genome size of about 8 kilobase, and encodes for 6 non-structural and 2 structural proteins. And the organization is very simple. There are no, unlike any segmented viruses, this is not a non-segmented virus. It codes for 2 major capsid protein, L1 and L2. And there are early genes. They are called E1 because they are expressed early in the virus life cycle. And this is called L1, L2 because they are expressed late in the life cycle. Now, let's look at what these proteins code for and what is their function. The protein E1, E2, and E4 are all involved in viral DNA replication. However, E2 has a very special function. It also controls or keeps E6 and E7 expression in check. So once the HPV infects, predominantly it stays outside the human nucleus. It stays as episome. It doesn't cause any problem. It just replicates and keeps shedding. That's not an issue as far as the disease is concerned. However, the problem comes HPV when it integrates. And when HPV integrates, it integrates from this loci. And when it does that, it loses part of L2, and it loses entirely the E5 and part of E2. Now, what happens is once it has lost E2, you can appreciate that it is going to lose control over E6 and E7 expression. We'll see what that means. Now, I had shown this cartoon earlier that there are 2 different critical checkpoints for cervical cancer. One is correcting the DNA damage and any errors that is caused in replication. And the other one is responsible for keeping the cell cycle in quiescence. Now, E6 specifically degrades, primes P53 for degradation, and it activates telomerase. When it activates telomerase, the cell cycle actually loses control, and with activation of telomerase, E6 has a potential of converting a technically quiescent cell line into a immortal cell line. I'll give you an example. HeLa cell line has been maintained in culture system for more than 40 years. If anybody out there is known to have propagated any mammalian cell, you would know that more than 200 to 300 passage, you would not be able to grow the cell further. The cells would lose its viability because the telomerase is no longer active, and it loses there is a chromosomal instability, and the cells die. They no longer propagate. So E6 potentially converts any cell line into an immortal cell line. So basically, that's what you want. A cancer is essentially an uncontrolled cell division. On the E7 front, E7 subjects retinoblastoma for degradation. The phosphorylated retinoblastoma sits on the elongation factor, preventing the cells to enter the synthesis phase. But once the phosphorylated retinoblastoma is degraded, the cell cycle loses its control. So what you see is there are two aspects to HPV infection. As long as the HPV is maintained episomally, as long as it is not integrated into the human genome or host genome, it's not going to cause any problem. It just replicates and sheds the virus particle. The moment it integrates, because some of the control mechanism of the virus itself has been lost, the cell cycle loses its control, and because there is no error-prone error correction mechanism, there is a genetic instability introduced, and this is perfectly suited for creating a cervical cancer. So how does E6 and E7 evade immune system? So first of all, HPV is present inside, it resides in keratinocyte. This is a site which is not actively patrolled by immune surveillance. Then also HPV actively goes on to impair antigen presentation. You see E6 and E7 block TLR either directly or indirectly. The dotted lines are all inhibitions. E6 can block including the interferon responsive transcription factor. It activates YAP, which is another transcription factor inhibitor that goes on to blocks TLR. And overall, you see E5 is directly involved in antigen presentation. So the virus has evolved a strategy that once it integrates into the host cell is not able to mount an effective immune response to this anymore. Now, if you were to look at how the cancer progresses, first thing is, fortunately, the infection from HPV and progression into cancer takes several decades. It's a very, very slow virus, and because it's a DNA virus. The first stage in cytological term, it's called low-grade intraepithelial lesion, LSIL, and HSIL is high-grade squamous intraepithelial lesion. Histologically, they are called CIN1, CIN1, 2, 3, which is cervical intraepithelial neoplasia. On microscopy, you would see this is how a normal cell polarity is. You have a basal cell layer, which is extremely nucleated, and as it progresses towards the surface, where the epidermis is, the cell is actually going to lose its nucleus and become flat squamous epithelial cell. And that's what is required to create a barrier. But as the HPV infection takes place, it converts these basal layer into transformed epithelium. They start dividing more and more. In the CIN1, CIN2 phase, there is moderate amount of cell division taking place. More and more nucleated cells are visible, and that's what Pap smear is looking for, how many nucleated cells are available. And the proportion of that would be used to grade CIN1, CIN2, or CIN3. So as the infection progresses over a period of time, you'll have only cells which have completely lost their genetic stability and has lost cell cycle control. The initial stage, as I said, is a productive stage where the virus is still being shed, but it's not still cancerous. So anything at this CIN2, there is a possibility of this reversing back to normalcy. That we'll see in the next slide. So what happens when there is an infection? Within weeks, you would see the first lesion. And by which time, if you were to sample the cervical cytology, cervical smear and look for DNA, they would all be DNA negative. And within few weeks to few months, you would see the first immune response is cell-mediated immune response. You don't see any antibody. But as it goes on, you'd actually see a very robust immune response triggering in, which is an antibody-mediated response, and that leads to almost 80 to 90% of the HPV infection gets cleared automatically. It's only the 10% of the population that actually goes on to harbor the HPV virus. As I said, the longer the virus stays, the chances for it to integrate into the host genome is higher, and the higher the chance, the more is the likelihood that you would create genetic instability into the host, and therefore that would cause for progression into cancer. So from the onset of infection to measuring a detectable immune response could take several months, but a mere infection to HPV is not enough. You need to have persistent infection. That is, the infection has to last for several years before enough genetic instability is created for the cells to migrate into a cancer. So again, it's the same thing. The persistent infection is what is the high risk. Important is the immune response is also how your nutritional status is. The poor nutritional status is also one of the risk factors for people to progress into cancer. And poor Th1 to H2 response, immune suppressant. If you are immune suppressed, you are more likely not to clear the infection and get into the persistent infection. Smoking is also attributed to be one of the major risk factors. However, there is more with Langerhans depletion. Now, people out there should not be scared that as long as you are able to do a periodic screening of cervical cytology, that itself is a very effective intervention. You can probably manage the disease very, very effectively. However, the problem is across the world, especially in the developing world and lower income countries, that periodic screening is what is completely missing. So, having spoken about what HPV does, let's look at what are the intervention strategies. As I said, the cervical cytology could be just under microscope. You could visualize that. That's called pap smear. Of late, there are DNA detection with PCR that's possible, but again, this is all cost and labor intensive. On the immunological intervention front, you could have vaccines, which is far more cost effective and that's amenable for public health administration. And you could have a prophylactic vaccine or a therapeutic vaccine. A vaccine would have, which promotes regression. That's one possibility, that it actually clears an infection. It's called therapeutic vaccine. In addition to that, the current practice of care is if the doctors see any lesion, they do undertake loop-mediated electrical excision of the tissue, or even sometimes hysterectomy to take away the diseased tissue out. However, that's actually not very good care to remove the entire uterus because there are other hormonal complications involved with that. Now, what led to vaccine as an intervention strategy is that when people took the serum from infected animals and they transferred it passively, that passive transfer alone could prevent the infection. However, when you use the heat-killed preparation of this lesion, then the infection could no longer be prevented. So what it told you is that you needed the viral coat proteins to elicit a protective immune response. But not only that, you needed the viral core protein to be present in a conformationally correct form. Now, what are the options available for vaccines? Typically, those who are studying vaccines would be taught that you could have a killed or attenuated strain of a pathogen. But in case of HPV, that is not possible. You cannot grow HPV in an industrial scale. You can grow for an academic purpose. It requires a very complicated keratinocyte graft culture, but that's not something possible to perform at an industrial scale. Well, you need to produce millions of those. The recombinant L1, L2 capsid is something that is already being deployed, and we are going to look at how this is made. And the other version is using E6 and E7. As I said, E6 and E7 are the key protein that are pushing the cervical cancer, the cervical tissue into cancer. So if you could vaccinate against E6 and E7, probably you could have a therapeutic version. But this is something still under development. We have not seen any positive outcome, although the L1 based vaccine is already out. Now, let's look at how the virus actually looks like. So this is how the cryo-electron microscopy of the virus, papillomavirus, would appear. You can appreciate this is a pentameric structure comprising of 5 L1. The L2 is buried inside these grooves. And because of that, it's no longer visible to the immune system. The immune system sees the L1, predominantly L1. The L2 is visible the moment virus starts invading the cell, when it is getting internalized. Only then the L2 is visible. Although L2 as a therapeutic prophylactic vaccine is also gaining some attention. So there are a total of 360 copies of L1 in one virus particle arranged into 72 pentamers, and about 72 copies of L2, because each of these pentamers contain one L2. Now, what was the serendipity? So when you cloned HPV L1 and expressed into a heterologous system, and this was in the early days, it was yeast, to their surprise, they saw that the L1 could assemble into virus-like particle. And this virus-like particle looks very similar to the virus itself. So body sees the virus-like particle as virus itself and mounts a very, very effective immune response. So why is this such a breakthrough? There is HPV, as I said, contains E6 and E7. It is difficult to grow. E6, E7 have the potential to immortalize any cell line. Therefore, handling HPV virus per se is going to be extremely risky as far as the operators are concerned. So if you have only L1, which has no oncogenic potential, and if the L1 alone can form a virus-like particle, you have a vaccine that can be produced at an industrial scale. But not only that, you have the technology that is extremely safe. So that's what happens. The L1s are cloned and they are produced in large fermenter. You identify the VLP under the correct folding of the VLP in EM, and you can perform several of the testing to say that, okay, we have a VLP. Now, as I said, that the HPV itself takes about, the establishment of cancer takes about 30 years. So how do you run a vaccine efficacy trial? You cannot have people vaccinated and monitor them for 30 years. So you needed an assay system, which would be surrogate assay system. So the pseudovirus particles have another property that they can encapsulate. L1 can form not only a virus-like particle, they can encapsulate anything that is 8 kilobases. So what John Schiller and all did, this is what is called a pseudovirus assay. They cloned a reporter gene which could be expressed in mammalian cells, and they mixed these L1 capsid and the reporter gene together to form a pseudovirus. It's called pseudovirus because it has, I'm sorry. It's called pseudovirus because it's containing a pseudogenome. It's not containing the viral genome. And once it infects, it expresses the reporter gene that can be measured. Whereas if you have an antibody, suppose you have a VLP vaccine, this antibody is going to neutralize the virus, and by virtue of its neutralization, there is no reporter gene transfer into the mammalian cell, and therefore you don't see an expression. So if essentially a neutralization antibody assay is a measure how you would say that my vaccine has worked. The clinical trials, therefore, are not run with a clinical endpoint. It is run with a virological endpoint wherein you screen for the presence of virus. Now, so there are several vaccines that are out there. Cervix and Gardasil were the first vaccine. Gardasil contains 4 HPV types, HPV 6, 11. These 2 HPV types can cause genital wart, whereas 16 and 18 cause cervical cancer. Cervix contains HPV 16 and HPV 18, and Gardasil is expressed in yeast, whereas cervix is expressed in insect cell. Gardasil is basically a Merck vaccine. It has come with another variant, which is a 7 variant variant, and this is also expressed in yeast. What is important is the red residues, amino acid residues, are something that's hypervariable residues, and they are type specific, meaning each type will have different residue. And whereas the blue residues are extremely conserved residue. So this is the side view, this is the top view, and this is the bottom view of the L1 pentamer. So this is what is exposed on surface. So what you see on surface is what you have type-specific residues that are exposed. And so HPV VLPs do confer immunity to specific type. That means if you have HPV 6, 11, 16, 18, it will definitely confer immunity to 16, 18. Whereas, although there are reports of cross-protection available. So how has this vaccine helped in public? So this is what you see in Argentina. This is a trial conducted recently and published recently. They used quadrivalent HPV vaccine, which is Gardasil. They had HPV 6, 11, 16, and 18. So you do see marginal reduction in 6, 11 cases, but you also see a significant reduction in cases of 16 and 18. So you look, black is unvaccinated population, and this is vaccinated population in the same setup. So when you vaccinate, you definitely prevent onset of infection to cancer-causing virus. So therefore, it's an effective intervention strategy. So to summarize, HPV infection is very common, and it occurs rampantly in higher, in low and middle income countries. Cultural and economic factors increase the vulnerability. Women in low and high middle income countries are not somebody who come forth asking or seeking help for their own health. That's another reason that burdens are very high. Nutritional status is definitely another reason. VLP vaccine is extremely effective. The current technology to produce vaccine is cost-effective, and safe. The approach is, unfortunately, the current vaccine platform is extremely expensive. So we are working towards a method or a platform which can minimize the cost so that the vaccine can be available to many and more people across the world. So if you have one or two questions, I'd quickly take those. And because, as you heard, there are several phone calls coming up, I have to rush in for another meeting.
Okay, Esther, is there any questions?
There is one question, sir. Does the vaccine contain the weakened virus, and do cervical vaccines give long-term results?
Yes, the HPV vaccine is made only of L1. There is no virus involved. You cannot grow the virus, and therefore it's extremely safe. There is no weak or no killed virus involved. It's a recombinant virus, VLP. It's called virus-like particle. And in terms of long-term immunity, yes. So far, people have not reported any outbreak of HPV infection in people who got vaccinated. So it does confer a very long-term immunity.
Okay, thank you, sir. There is one more question.
Yeah.
It's really antibody. Will the antibodies really suppress the virus?
I do not know what suppression meant. What the antibody, if there are neutralizing antibody, they definitely go and neutralize the virus from invading an epithelial cell. So as I showed earlier, in the productive phase of the HPV infection, the virus particle is still being shed, and you could, if there is neutralizing antibody,
Yes, sir. Sir, there is one more question in the chat box. Could you please tell the right age for taking a vaccine?
The recommended age is between 15 to 19 years. That's the adolescent vaccine. And but I think you could still take the vaccine if you have missed that age.
Yes, sir. So another question has come up, if you have time for that. Will the vaccine protect women from cervical?
Can I just take a call? Just this
Sure,
is
sir.
urgent.
Sure,
I'll
sir.
answer your question.
Okay, sir.
Yeah, I'm on a call. I just call back. I know Sashi is calling me. I'll call back. Yeah.
Okay.
Yeah.
Okay. Will the vaccine protect women from cervical cancer who have already been exposed to HPV?
No, it's a prophylactic vaccine. That means you need to take before the virus has hit you.
Yes. Thank you
And
so
the virus infection happens, as I said, the moment you have any intimate contact.
Yes, sir. Okay. Thank you so much, sir, for your time and for giving us an insightful knowledge into the world of cervical cancers and HPV. We know you are a very busy man with another call on the line. So I would like to invite our Honorable Vice Chancellor to say a short speech of appreciation about the thanks.
Thank you, Dr. Rajan, for your excellent lecture. And it was a wonderful, because Mizoram is a place where we have a lot of incidences of cancer. That is the reason why I was requesting you. It is almost considered to be highest incidence of cancer in Mizoram. And I was actually asking my biotechnology faculty to have a collaboration with your Reliance, and we can have some kind of collaboration in planning for cancer vaccines. And also I was also talking about COVID vaccines. They already started initiated some work. So industry collaboration with our university will be very good. So our people are our biotechnology department is very vibrant department. So you plan for some kind of collaboration, because being a vaccine head, you can collaborate with our university. This is what I wanted to request you. And that is the reason why I invited our biotechnology department to have organizing secretaries for this particular webinar. and also to have a collaboration in future. So
Sure.
so with your experiences, you can guide us to start some kind of work. And if you find under BIRAC program or some other program, we can apply some projects together with our Mizoram University. This is what I wanted to request you, and also in future I hope that we can work out some good research work. Thank you.
First of all, thank you very much, Professor Rao, because it's an honor to be presenting to you and to your guest faculties and students. We at Reliance are open to looking at several possibilities, and I'm open to talk to you, and we can discuss these offline. And collaborating with academic institution would be definitely one I would do. You know that we did that in engineering also earlier. So we are open to these kind of academic interactions. So we keep the option open.
Okay.
Thank you. Thank you very much.
Thank you all. I want to thank our Honorable Vice Chancellor for giving us this time to conduct this webinar. I would like to thank our speaker again for giving us, such incredible knowledge. And I would also like to thank everyone who has participated here today, including all the technical staff who made this possible. So I wish you all have a good day. Thank you for attending this webinar.
Thank you for conducting it so well and on time. I'm impressed. Yes, thanks.
Thank you.